My name is Isaias Martinez, and I'll be presenting on our project, Liposome-stabilized WNT3A Protein upregulates adult hepatic stem cells and reduces cell death in liver during cold preservation. As the demand for liver transplants continues to grow, the transplantation of extended criteria livers becomes more crucial to address the organ shortage. These livers, often sourced from older or high-risk donors, are more prone to ischemic injury during retrieval and storage, leading to reduced quality and viability. This can result in suboptimal patient outcomes and increased risk of transplant failure. Our team aimed to explore the potential of L-WNT, a potent activator of adult hepatic stem cells, as a preconditioning agent. The objective was to investigate whether L-WNT could reduce cell death, increase stem cell activity, and improve liver quality before transplantation. Our research consisted of both in vivo and in vitro experiments. In the in vivo study, we worked with heart-beating wild-type mice, injecting them with either L-WNT or saline via intraportal administration. This allowed us to evaluate the immediate effects of L-WNT on liver tissue. After 15 minutes, we retrieved the livers and stored them at a cold temperature between 0 and 4 degrees Celsius for 2 hours. This simulated the typical storage conditions for transplant organs. We then assessed apoptosis using tunnel staining, a method that identifies DNA fragmentation associated with cell death. This gave us a clear indication of how L-WNT influenced liver viability. In the in vitro experiments, we focused on liver slices, treating them with L-WNT to determine its effects on stem cell activity and apoptosis. We specifically measured the number of apoptotic cells, evaluated caspase-3 expression, and compared these metrics to those from slices treated with saline. This approach allowed us to understand the impact of L-WNT on cellular health and stem cell activation in a controlled environment. The results from our in vivo study were quite promising. We found that preconditioning with L-WNT significantly reduced the number of apoptotic cells compared to the control group, indicating by a significant decrease in tunnel staining. This suggests that L-WNT can protect liver cells from ischemic damage, which is critical for the viability of transplanted organs. In the in vitro study, L-WNT treatment led to an even more pronounced increase in the number of WNT-responsive cells, indicating a strong upregulation of adult hepatic stem cells. Additionally, the number of apoptotic cells and caspase-3 expression levels were significantly lower compared to the control group. This data supports the hypothesis that L-WNT can effectively reduce cell death and enhance stem cell activity in liver tissue. Our research demonstrates that preconditioning livers with L-WNT can be an effective approach to reduce cell death and enhance adult hepatic stem cell activity during cold storage. These findings are significant because they suggest that L-WNT could be a valuable tool for improving the quality of organs before transplantation. Ultimately, this could lead to better post-transplant outcomes for patients with a reduced risk of organ failure and improved long-term survival. Given the positive outcomes, our team plans to continue exploring the potential of L-WNT in liver preservation. The immediate next steps include evaluating various preservation conditions, such as different storage temperatures and durations, to find the optimal range for liver viability. We will also examine a variety of preservation media compositions to determine the best conditions for long-term organ storage. Beyond these evaluations, we aim to study various concentrations and timings of L-WNT to establish the most effective dosage for reducing apoptosis and enhancing stem cell activity. By using animal models, we can refine our approach and identify the best practices for liver preconditioning. This research could ultimately inform the development of new protocols that improve liver transplantation success rate and patient outcomes. Thank you.